In this presentation, I would like to give you some information about ELISA Kits in BioVendor portfolio, which are intended for assaying of pulmonary disease markers, or more generally, protein molecules, somehow participating in pathological processes that occur in lungs. BioVendor company has a long tradition in the development of immunoassays, as well as the development and production of antibodies and recombinant proteins. Our ELISA kits for the determination of biomarkers in various clinical fields have gained a solid reputation and were frequently referred in top-class scientific journals. Here you can see only short extract from all ELISA kits that BioVendor offers from the field of pulmonary disease. I will be speaking about only some of them in detail, those marked with bold letters, which are more relevant in diagnosis and research of pulmonary disease. Before I start with the first protein molecule, let me remind types of samples which are suitable for pulmonary disease markers measurement. We can test, of course, serum or plasma samples, or there is a possibility to measure markers secreted in respiratory tract. So we can use, for instance, sputum, bronchoalveolar lavage fluid, or similar fluids. The first molecule we have selected for you is club cell protein, also called CC16. It was formerly called also Clara cell protein, but using this name was suggested to be stopped a few years ago. The German scientist who discovered and described pulmonary club cells, where club cell protein is primarily produced, Max Clara, had been a member of Nazi Party and used for his research tissue samples from executed prisoners during the Second World War. Human club cell protein may be considered as a marker of epithelial airway injury. One of the main functions of club cells is to protect the bronchiolar epithelium. In this context, CC16 has been largely studied, especially in association with its protecting effects at cigarette smoke-induced inflammation and injury, and in chronic obstructive pulmonary disease, COPD. As you can see in the picture, active smokers have significantly reduced serum levels of CC16 compared to non-smoker controls. COPD patients, this group comprised only non-smokers, have even lower CC16 levels in serum than smokers. Reduced levels of CC16 may mean also increased risk of chronic bronchitis, and they are also associated with cystic fibrosis, pulmonary exacerbation. CC16 has been indicated as a potential therapeutic target for COPD. Now, let's take two proteins together. These are surfactant proteins, A and D. They form a principal part of the protein component in the lung surfactant and participate in innate immunity response. SPD serum levels are elevated in patients with idiopathic pulmonary fibrosis, IPF, and associated also with increased mortality. SPA might also serve as a predictor of mortality in IPF. This protein is also potentially useful as a biomarker, which can help to differentiate among various forms of interstitial lung diseases. Both these proteins have been evaluated in extensive meta-analysis using big number of studies as useful for differential diagnosis and prediction of survival at IPF. SPD is further supposed to be one of the best markers of COPD, especially in a combination with certain other markers. KL6 is highly glycosylated protein, which was first suggested as a tumor marker. Later, it appeared that it is inferior if compared with some other cancer markers, like carcinoembryonic antigen CEA. KL6 promotes migration, proliferation, and survival of lung fibroblasts. It was suggested as a diagnostic marker generally for all interstitial lung diseases, specifically as a diagnostic and prognostic marker for idiopathic pulmonary fibrosis. Its increased levels might mean also increased risk of idiopathic interstitial pneumonia, and it was used as a diagnostic marker in sarcoidosis. Further, connective tissue growth factor CTGF is a fibrogenic marker for chronic fibrotic diseases. We can find increased CTGF concentrations in blood and urine of patients with idiopathic pulmonary fibrosis and in blood of patients with stable asthma. CTGF is a central mediator of tissue remodeling and fibrosis, as is shown in the picture on the right. It participates also in fibrotic processes associated with radiation therapy and cancer treatment. That is why it might be necessary to monitor CTGF concentration as a marker of these fibrotization processes. BioVendor offers also ELISA kits for determination of some matrix metalloproteinases, namely 3 of them, MMP7, MMP8, and MMP9. I put just a couple of general pieces of information about these proteins. They are proteases with zinc binding motif. It means they have enzymatic activity, due to which they are capable to degrade extracellular matrix, and this way to participate in tissue remodeling or maintenance. MMPs regulate release or activation of various important molecules, like cytokines, chemokines, hormones, etc. They are associated with processes like immunity response or inflammation. This list shows the discussed ELISA kits with their respective catalog numbers. From MMPs, only MMP8 is mentioned here. BioVendor can offer you also a recombinant, prokaryotic or eukaryotic proteins concerning pulmonary disease research and/or diagnostics. In addition, we offer also some antibodies applicable in this field. I believe you have found something interesting in my presentation, also for your own work. But it was just a small starter from a wide menu of BioVendor biomarker immunoassays. Visit us at the BioVendor.com website, where it's quite easy to find an ELISA kit, protein, antibody, or Mirna immunoassay meeting your needs. Feel free to contact our product managers and ask for help.